Assalamualaikum and salam sejahtera to everyone. I am Farahin and my team members are Azmira Alia and Siti Shaida. We are from Group 3. Today we will be presenting about the given data for our final lab assignment. The introduction. In this lab assignment, two methods was used, which is PCR and agarose gel electrophoresis. PCR or polymerase chain reaction is a method used to amplify a specific DNA region, while agarose gel electrophoresis is a method to analyze and separate DNA fragments based on their size and charge. So once the PCR is completed, gel electrophoresis is done to visualize and analyze the success of the reaction. In gel electrophoresis, we can see that the smaller DNA fragment will travel further compared to the larger DNA fragment due to the mesh-like structure of agarose gel. DNA extraction is also one of the lab procedures done in genetic engineering. So what is DNA extraction? DNA extraction is mainly done to isolate the DNA from the nucleus of cell. The successful gel electrophoresis and PCR is determined by the efficient cell lysis and also the quality of the recovered DNA. So, to have a successful extraction of DNA, few processes are involved, such as the disruption of cells, the denaturation of protein and nucleoprotein complexes, the inactivation of nucleases such as DNAs, and also the recovery of the DNA. The extracted DNA should have low contamination of proteins, carbohydrate, lipid, and also RNAs. This is because the result of the experiment will directly affected by the quality and the integrity of the isolated nucleic acid. It's very likely that PCR will play a major role in future molecular diagnostic technique. For example, the applications can be seen in microfluidic PCR-based system and also in droplet-based quantitative PCR. In commercial product, PCR-based detection systems are already available for the detection of bacteria and viruses on chip. Due to the COVID pandemic, it speed up the miniaturization of PCR to move toward truly portable sample-to-answer system for point-of-care application.
Now, let's move on to the result. I am going to explain the result for crude DNA extract first. The quality of crude DNA extracts was different between the four environmental samples. First, length 1 to 3 activated sludge. What we can observe from length 1 to 3 is the DNA band with a low level of impurities. Second, from length 4 to 6 river sediment, we can see that the DNA band still visible but got slightly smearing above of DNA bands. Last but not least, for length 7 to 9 and length 10 to 12, anaerobic digestion sludge and nitrifying sludge respectively, all of the length, the result is distinct DNA band but high background smearing, which the level of impurities is high. And also, what we can see at the length 10 to 12, each well DNA tried to come out of the well, but some DNA still remain inside the well. Next, result for PCR amplification by using universal PCR gel. At length 1 to 3, where we can see that DNA band looks good and sharp but slightly smearing, so that the PCR amplification result is positive. While for length 4 to 12, got no distinct DNA bands produced. Thus, the PCR amplification result is negative. Overall, what we can conclude for the result is positive PCR amplification was only obtained for length 1 to 3 activated sludge, while another length is failed.
From these gel images, significant smearing can be observed. Now, what causes these smearings can range from unsatisfactory gel preparation, from having too much sample analytes, or even poor quality sample. A poorly prepared gel is uneven and could yield smeared results, whereas concentrated samples will contain particles too large for the wells and produce smearing. From our observations, it could be deduced that the contamination of proteins or high salt concentration, degraded or denatured samples may have contributed to the smearing. Careful preparation of both the gel and the sample minimizes the possibility of smearing and yields clear bands ideal for scientific interpretation. From what we can observe from the gel electrophoresis of the crude DNA extracts, it is found that the activated sludge had the least amount of impurities, followed by the river sediment, and the anaerobic digestion and nitrifying sludge both contained high levels of impurities. This was deduced by the presence of distinct bands and level of smearings observed. In order to prevent contamination of DNA extract that yields poor results, precautionary steps must be taken. This includes designation of areas and tools specific for tasks such as extractions and amplifications. Remember to use gloves and always assemble a negative control reaction. Moving on to the success of the PCR amplification, there are many factors that could contribute to either the success or the failure of the experiment. This includes the purity of genomic DNA, the length of DNA fragments, the precision of DNA concentration measurements, the presence of single nucleotide polymorphisms, the uniqueness of PCR primer sequences, the concentrations of the PCR buffer reagents, the primer length and the GC contents of primers and template, simple repeats in the primer sequence, and stable secondary structure of both primer and product sequences. Hence, many different scenarios can contribute to the outcome of this experiment. From the gel images of the PCR amplification, it is found that only the activated sludge had a positive PCR amplification, whereas the rest failed. This could be explained by either the purity of the genomic DNA or the use of universal PCR set. From the analysis of the crude DNA extract earlier, the activated sludge showed least amount of impurities, which could have led to it being the only successful PCR amplification. There were high levels of impurities for all the other samples, which may have disrupted the PCR process. Another factor could be the use of the universal PCR set. Universal primers and restriction enzymes are theoretically complementary to a large fraction of gene sequences in a database. This does not necessarily mean that the primer or enzyme are optimal. Sequences in the database may be incomplete or corrupt, and most databases do not have sufficient genetic data. This reduces the specificity of the primer and enzyme, hence could cause PCR failure. Now, how do we solve this problem? Since we have made a precursor gel electrophoresis of the crude DNA extract, early detection of the sample contamination could prevent further amplification reaction. Once a crude DNA sample was shown to have high levels of impurities, DNA extraction and isolation has to be redone in order to correct this error. We should also try to use gene-specific primers and restriction enzymes to improve the PCR results. This is because these primers and restriction enzymes are specifically designed and meant to be used on the targeted DNA. This could be an improvement rather than using universal primers and restriction enzymes.